Thank you for joining me for Lesson 7 of the Biochemistry Literacy for Kids Teacher and Student Training video series. This lesson is all about amino acids. And we actually know a little bit about amino acids already. We know that they are acids, which means that they're acid-base active. So what does that mean? It means that their exact structure is affected by their environment. So if we have a very acidic environment, like we see here, we're going to have a completely protonated amino acid. That means that the nitrogen is going to be positive charged, and it's going to have 4 bonds. 1 extra proton is added, so that's the acid form. And also the oxygen on the right side here is going to also be protonated. It's going to have 2 bonds. That's the acid form of that group on the right side there. As the amount of protons in the solution decreases, the amino acid actually can sense that, and its protonation state will change. So as the amount of protons go down, we actually have the ability to lose a proton on that oxygen. If the amount of protons goes down even more, the nitrogen on the left side will become deprotonated and become neutral again. And this shows us that we now have the double base version of what used to be the acid. So we've seen all this before. We've seen how oxygens and nitrogens can be acid-base active. This connects to the concept of pH and pKa. The solution that I'm showing here would be a low pH, a very acidic environment. This would be more of a neutral environment, and this would be more of a basic environment. But we'll have a lesson in the future that kind of concentrates on the acid-base idea. This is really the most common form that we're going to see amino acids take. The form that we see is what's called the Zwitter ion, which means that we have a mixed ion. We have a positive charge on the left side, negative charge on the right side. Usually when we draw amino acids, which is what we are going to start doing in this lesson, we are going to draw this particular form of the amino acid. And that's because we tend to use amino acids in a neutral solution. Cells and biology tends to be in a neutral solution. So we're not usually going to find the amino acid in this form or the amino acid in this form. This is the most common form. So as we draw amino acids later in this lesson, I'm going to encourage people to draw the Zwitter ion, which is this particular form, the mixed ion, showing that we have the amino acid in a neutral environment. So besides the acid-base nature of amino acids, they also have another quality which is really important, and it's important for building a biochemistry literacy, and that is the concept of chirality. So you may have seen a little bit of this before in some of the lessons. Sometimes the bonds in a molecule are drawn as wedges or these dashed wedges, which you're seeing here. So what is this about? This is about whether the bond points towards the observer or away. So we can kind of see here that if we look at that central carbon, that there's one bond that is kind of pointed more towards us and one that's definitely pointed away from us. That solid wedge is meant to show one of the bonds is pointing towards us, and that dotted wedge is meant to show that that hydrogen is pointing way back into the screen. That is the basic idea of chirality. We're going to talk a lot about chirality, and it's going to become way more familiar to you in this lesson. But by the end of the lesson, you're going to really, it's going to be very clear what these wedges are all about. So the place that we've seen these wedges before was in the flavor molecule unit. In particular, the molecule of menthol has some of these solid wedges and a dotted wedge drawn. So what this means is that some of these bonds are pointing towards us, the top 2 bonds, and some of the bonds are pointing away from us. And when you build the structure, you really can see that you'd have to make a choice whether the bond points towards us or away from us. This is where the models can become very helpful. We're going to do a lot of model building in this lesson, and also we're going to look at the molecules in PyMol. So we're going to get very familiar with this concept of chirality. It's not going to be a big deal by the end of the lesson. The other name for chirality is molecular handedness. The word chiral comes from the Greek word for hand, actually, and there's a few other words in English that have this chiro sort of prefix to them. We have chiropractors, which use their hands to manipulate the spine. We also have the order name for bats, which is Chiroptera, which means hand wing, right? Chiroptera is a hand wing. So this idea of chirality has to do with handedness. Now, before we start studying chirality, molecular handedness, I want to show you some molecules that are not chiral and are not handed. This molecule is ethanol. I drew two of them here. You'll see why in a second. Ethanol isn't handed. The technical way of figuring out whether a molecule has a left or a right hand, whether it's a handed or chiral molecule, is that you have to look at all the atoms and decide whether any of the atoms have four different groups or different patterns of atoms attached to a single atom. So if you just read at the bottom here, it says it does not have a carbon with four different groups attached. We're going to see what that means in a minute. But if you look at any of these carbons, we see a lot of repetition, right? We see a carbon that has 3 hydrogens. Those are not all different. We see a carbon that has 2 hydrogens. Those are the same. So this is not a chiral molecule. This idea of chirality is going to make sense when we look at this next example here. Here's a different version of ethanol. This is fluoroethanol, 2 different versions of it. And these are chiral molecules. These do have a handedness to them. And again, that technical reason of the technical definition is that a chiral molecule, does the carbon have 4 different groups attached? So can we find an atom, usually it's a carbon, that has 4 different groups attached? So let's look at the 2 carbons. First, we have the carbon that has 3 hydrogens. That's a pretty boring carbon. 3 of the same hydrogens does not make it chiral. But this other carbon is a very interesting carbon, and it has 4 different groups attached. One of the groups is a hydrogen. One of the groups is a methyl group, a carbon. One of the groups is a fluorine, the green one. And one of the groups is the oxygen, the alcohol part. This fits the criteria for chirality. It's a carbon with 4 different groups attached. Now, some people may be getting a little bit worried. From my experience, this chirality concept is a little bit confusing. What's he talking about? Left hand, right hand, chirality, 4 different groups attached. Let's look at it in an even simpler way. Let's remember that the ethanol molecule looks a little bit like a dog, right? It's like a little doggy molecule. I'm going to give you a little analogy here. Let's look at the dog and imagine that it has a left hand and a right hand. And whatever hand is white, that's the hand that it uses to write. That's its handedness. So if the dog has a white left hand, that means that the molecule is left-handed. If the dog has a white right hand, it means the dog is right-handed. So now can you tell that these are different molecules, that it's a handed molecule? The molecule on the left has the white hydrogen on the left, so that's actually the left-handed molecule. The molecule on the right is right-handed because it has a white hand on the right side. So the interesting thing here is that it proves that even though these 2 molecules look very similar, that they're actually completely different. They're not the same molecule. If you have someone who's right-handed and someone who's left-handed, those are different properties of those people. They're actually different people. Same thing goes for molecules. If you have a molecule that has very similar connections, but one is a right-handed version, one's a left-handed version, these are really different. And we've seen before that many molecules, if you just change one atom around, it completely alters what the molecule is. This is another example of that idea of isomerization, of the isomer, that something can look very similar, but if the bonds are a little different, it's a different molecule. So again, finding that carbon that has 4 different groups attached is really important. We tend to write a little star on that chiral carbon, the carbon that indicates that we have a handed molecule. And there's just a few slides here that have different ways of going through this explanation. How do we know these are 2 different molecules? You can kind of just view this in different ways. Just create a few different forms of the molecule just so you can practice this. The idea here is that don't worry about whether the molecule is positioned one way or another, if it looks one way on the page or the other. You have to look at where the white arm is. If the white hand is on the left side, it's just a left-handed molecule. Doesn't matter which way the molecule is drawn. If the white hydrogen is on the right side, it's got to be the right-handed form of the molecule. And then here's just a little puzzle here. It's 4 different drawings, but there's actually only 2 kinds of molecules here. And if you just look at the molecule as though it was a dog, you would see that there's really only two kinds of molecules. One just happens to be flipped to the other side, right? So the green ones, the left-handed ones, they're the same molecule, even though they look different drawn on the page, because it's a three-dimensional thing and we can flip it over. It's just the same thing. And it really just totally makes sense if you look at the molecule as a dog. Let's go ahead and look at the molecules in PyMol, because that can also help us out. All I'm really showing here is that, okay, so which one do we have here? Do we have the left-handed dog or the right-handed dog? It's obviously the left-handed dog. Its hand is white on the left side. So that's the left-handed dog, right? Is that the left-handed dog? Yeah, it's still the same dog. It totally makes sense. But if you were to draw this in an organic chemistry way, you would have, you'd maybe use the dotted wedge for the hydrogen this way, and then you would use the solid wedge this way. So I'm just kind of showing you that it doesn't matter in chemistry whether you draw it one way or the other. They can be the same molecule, the same structure. Now we're going to do this in a more formal way in a minute, but if this is still seeming a little bit weird, don't worry. This is no longer K-12 concept. This is a real college concept here, this chirality concept. You will not find this in high school. So it can be a tough thing to visualize, but I think with the model building, with using PyMol, and with continued examples here, this will really work out. You really will start to understand this chirality concept. Let's go through some formal steps to figure out chirality. Now, you won't always have a molecule that looks like a dog, that looks like it has a right and a left hand to it. So there's actually a more technical way of figuring out left or right hand. This dog kind of analogy here was just a kind of a little intro. So here's how you, in general, find the chirality of a molecule, whether a molecule is considered left-handed or right-handed. And by the way, it doesn't mean that it makes a person left-handed if they have the molecule, or if it doesn't make the person right-handed. It's not connected to actual handedness, okay? This is just a way of describing tiny structures like molecules. We can also have right-handed shells. We can also have right-handed spirals. There's lots of other things that can have handedness in the world. Molecules are just one of the things that have a handedness. So let's figure out the more technical and more in-depth way of figuring out handedness, the more general way. Step one, find that chiral carbon, that carbon that has 4 different groups attached. We did find it, okay? So that's something that you can easily do. Find a carbon that has 4 unique groups attached, no repeated groups like that. Other carbon has 3 hydrogens. That's not a chiral carbon. That does not contribute to a handed molecule. All right, step 2, this is going to sound a little bit weird, but I want you to go through it. Point the smallest atom, which means the lightest atom, away from you. And I've already done that for some of these molecules, but to really do this, you want to point the smallest atom away from you, and that's usually going to be the hydrogens. So we're going to get rid of those bottom ones because that's not going to help us. Those molecules are drawn in a way that it's not going to help us figure out the chirality in this kind of this easy way that most people do it. And we already know that they're duplicated molecules, right? We already know that one of them on the bottom is the same as one on the top, and the other one on the bottom is the same on the top also. So we don't actually care about those. We're just going to kind of forget those exist because they're duplicated. Step 3, we're going to rank the atoms according to how heavy they are. All right? That means knowing your atomic weights. So we know that the hydrogens are the smallest. What's going to be the largest? The fluorine. Fluorines are number 9, right? Hydrogens are number 1. That's going to be part of how we do the rankings here. The rankings are going to be the way we figure out the handedness of the molecule. This thing with spinning, I'm going to show you what that means in a second. Okay, so we're going to zoom up and look at this version, and we're going to do this ranking thing that I just mentioned a second ago. So what's the smallest atom, the lightest atom attached to the starred chiral carbon? The smallest one's obviously the hydrogen, number 1 on the periodic table. What's the heaviest atom? The heaviest one is actually the fluorine, number 9. It's smaller in this picture. We're going to see in a later lesson why fluorines are actually physically smaller than maybe like a carbon or an oxygen. But don't let that fool you. Fluorines are definitely heavier. They have 9 protons. So that's the heaviest. That's the highest priority atom that is in this ranking system. What's the second highest ranking atom? If you had to rate these in terms of how heavy each atom was attached to the chiral carbon, would be the oxygen, right? Oxygen is element number 8, so that's right next to number 9. What about the carbon? The carbon is element number 6, so that's going to be the 3rd heaviest element, and the hydrogen is still back there at number 4. we've now ranked the 4 atoms that are attached to the chiral carbon. This is all we need to do to figure out the chirality, whether the molecule is left-handed or right-handed. Now, we already know, because I created this tricky example here, that this molecule is going to be left-handed because it has the white hydrogen on the left hand. But if we were to kind of spin the molecule in the order of the top 3 heaviest atoms, the fluorine, the oxygen, and the carbon, we would see that the molecule would spin to the left. Okay? 1, 2, 3. If you count 1, 2, 3, it's going to the left. That's how you actually tell whether a molecule is left-handed or right-handed. It's this ranking idea. So this is just a cute example where I gave the dog the left hand that's white, but that's actually how you figure out the chirality in general. Let's keep practicing, because I know this is a weird concept. Let's look at the other version of the molecule. We already know that this is the right-handed one because it has a white hydrogen on the right hand. But how do we actually figure this out in a more scientific way? We're going to rank the atoms that are attached to that central chiral carbon. The easiest one to do is the smallest one. And remember this rule, the rule number 2 you can see here, point the smallest atom away from you. That's really, really important. When you build, and I hope you're building this model right now, build the molecule and point the smallest group, usually it's a hydrogen, away from you, and then focus on the other 3 atoms in the ranking. And those are the 3 atoms you use to figure out whether it's spinning to the left or right, whether it's left-handed or right-handed. All right, so again, we know that the fluorine is going to be the highest priority. It's the heaviest atom. Oxygen is the next heaviest atom, and then carbon's the next heaviest. So if we were to just look at the top 3 atoms, we would see that they rank, or they kind of spin to the right. And we kind of, in organic chemistry, will kind of spin the molecules like this, but the molecules are not actually spinning, although they are tumbling and rotating and doing things. Just because the molecule's chiral doesn't mean that it spins or doesn't spin. It's just kind of like a mental activity here, like a mental exercise to figure out whether we're going left-handed or right-handed. So here are those 4 steps to figure out handedness. Number 1, find the chiral carbon. Find a carbon that has 4 different things attached to it. Not all molecules have a chiral carbon or a chiral atom. So first figure out if the molecule is chiral at all. If it does have an atom that has 4 different groups attached, the first thing you're going to do is rotate the molecule in a way so that the smallest atom is pointing away from you. Now, sometimes I think of this as like a steering wheel. So you take the smallest atom, the smallest bond, and kind of hold it as though it was the steering column of the steering wheel, so that the 3 highest atoms are the steering wheel itself, and the hydrogen is like the steering column. So I hope that makes sense. Once you've done that and you've pointed the smallest atom away from you, then you're ready to rank the remaining 3 atoms. So figure out which is the heaviest, which is the lightest atom. That is going to help you figure out whether we are spinning to the left or to the right. If you count up your rankings, do you have to kind of circle to the right or circle to the left? That's telling us which of the chiral molecules we have. Is it the left-handed or the right-handed molecule? All right, so now we're going to try this again. If this doesn't make any sense yet, we'll do it to another molecule, and we'll figure out the chirality of a couple more examples here. All right, so I'm already highlighting the chiral carbon. This is our old friend here, alanine, a famous amino acid. The central carbon is chiral. We can tell that it's chiral because it has a hydrogen attached, it has a nitrogen attached, and it has 2 different carbons attached. Now, those are not the same groups, right? One carbon is a methyl group, it's a CH3 group, and one carbon is actually a carboxylate group, a COO group. That's the criteria for a chiral molecule. It's a carbon that has 4 different groups attached. Now, even though there's 2 carbons, they're still different carbons. They're not the same kind of group, right? So it fits the qualifications for a handed molecule. That's step 1, figuring out that we do have a handed molecule. What's the next step? The next step is point the smallest atom away from us, kind of like the steering column of a steering wheel. And I've already done that for you in this diagram here. We're now going to start ranking, right? So the nitrogen is obviously the heaviest atom. Nitrogen is number 7 on the periodic table. we have a tie, right, with the carbons, and we have the hydrogen as the smallest, right? So hydrogen is obviously the smallest, but we have to deal with this tie, and we have to figure out how to break the tie. So many people will figure out how to break the tie on their own, and this is actually the same way that scientists break the tie. Any thoughts on how we could break the tie there? We have two carbons. Which one is like the higher ranking or the superior carbon, right? Is it the carbon that has 3 little hydrogens attached? Or is it the carbon that has 2 big oxygens attached and one with a double bond even? The rankings are this way, right? The number 3, the kind of lower ranking carbon, is just the regular old methyl carbon. But the higher ranking carbon is kind of elevated to position number 2 because it has 2 higher ranking atoms to it, 2 heavier atoms. So I'm not going to get into all the rules of figuring out chirality, but it's basically the idea is that the higher ranking group beats out the lower ranking group. So that's how you figure out the handedness of an amino acid. So which way does this spin? If we were to build this molecule, and I hope you are building this molecule, and then figure out which way the rankings rotate or which way they spin, you would see that counting 1, 2, 3, that's going to give us a left-handed way, all right? It's going to be a counterclockwise way. If you were driving a car and this was your steering wheel, you would have to be making a left-hand turn, right? Going 1, 2, 3, that's making your car turn to the left. So this is a left-handed molecule. We can pretend that it's spinning to the left, and if you build the molecule, you can reinforce the idea by spinning the molecule to the left. That's a left-handed amino acid. Turns out that this is the kind of amino acid that we're made of. We are made of left-handed amino acids, not right-handed. If you looked at any of the proteins in our body, and the next few units are about proteins and peptides, this is the form of it that you would find. You wouldn't find the right-handed form, which is this one. All right? Again, we have the chiral carbon. Since we already know the rankings, I'm just going to pop these up here again. How do the rankings go here? If we had to spin the molecule in a certain direction, if we count 1, 2, 3, that's going in a clockwise direction now. If you take a steering wheel and turn it clockwise, which way does your car go? Turns to the right. This is the right-handed version of alanine. So alanine amino acid has 2 different forms. It has the left-handed form and the right-handed form. And we're now able to figure that out. Even though this doesn't look like an animal that has a left and a right hand, we can still use the ranking rules to figure out whether we have a left or right-handed molecule. So it's really, I think it's pretty amazing that the molecule on the left here, the left-handed molecule, that's what we're made out of. That's what life has chosen to construct proteins from. The molecule on the right is much, much less important in biology. This is not what we're not made of right-handed amino acids at all. There are a few examples where right-handed amino acids can be found in biomolecules, but for the most part, left-handed molecules, left-handed amino acids anyway, are what we find in biology. So even though a right hand and a left hand really look the same, we know that they're completely different things. A right-handed glove doesn't go on a left hand, right? And vice versa. That's very similar to how right-handed and left-handed molecules work. Left-handed amino acid is what our proteins are made out of. Right-handed amino acids are not really found in proteins, in at least in us. They have completely different uses, just like left and right hands. Now, we don't always have these nice three-dimensional structures to figure out chirality with, so chemists had to invent a nomenclature to show which of the bonds are more pointing towards us, which are pointing away from us. This is the key to helping us figure out, where those 3 higher-ranking groups are, so we can figure out whether it's left-handed or right-handed. So I'm going to show you how we transition to that. So I'm overlaying here a regular drawing of the left-handed, right-handed amino acid, and notice the hydrogen, right? See how the hydrogens are pointing down here, and they have a dotted bond to them? That's indicating that the lowest priority group, which is the hydrogen, is pointing away from us. So the remaining 3 groups are more pointing towards us, especially that carbon there. These 3 groups are the ones that you're going to use to figure out the ranking. So you could now look at either one of these, figure out, okay, nitrogen is number 1, the carbon with the oxygens is number 2, the carbon without the oxygens is number 3, and figuring out left or right-handed. But the main thing I want to show you here is just the idea of the nomenclature. The solid wedge is meant to show that the bond is coming towards us. The dotted wedge is pointing away from us. And you're going to see this a lot throughout the rest of my course and also in general as you study chemistry. And this is the idea of chemical and biochemical literacy, just knowing what that symbol means, knowing what those different kinds of wedges are about. One thing that I want to emphasize here is that usually when we draw amino acids, it's traditional to eliminate or just to just to omit the hydrogen, the lowest ranking hydrogen. Just makes it kind of cluttery. So it's just common to see the amino acids without those hydrogens drawn. Doesn't mean it's not there. It just means that we get rid of it just for simplicity's sake. But that hydrogen that's missing is always going to be the low ranking one, and it's always going to be the one that's pointed away from us. So you have to kind of either draw it in if you want to see it, or just imagine that it's there. In the next couple slides, we're going to start learning about the amino acids, and I'm showing this here now because I'm not going to be drawing that little hydrogen. It's going to be omitted, but it's not that it's not there. It's just that it's not traditional to show it. Okay, as we said before, almost all amino acids in humans are the left-handed ones, and those are going to be the ones that we're going to be studying right now. So this is going to be a table of the 20 amino acids that make up us, the 20 amino acids that are part of the proteins and enzymes that do most of the interesting biology that makes up life. All right, so we're going to fill in this whole chart here, and this is part of the worksheet, so you should have this. But we already know one amino acid because we've been studying it for a couple lessons now, is alanine. Alanine is represented by the code A. For shorthand, we're going to call alanine A sometimes. That's the alanine molecule. Let's go through and just quickly introduce all the 20 amino acids. There's a simpler amino acid that is called glycine, and this looks just like alanine, right? But it has a missing carbon. That third carbon is not there. There's only 2 carbons. That's a glycine. Now, before we continue looking at all the other amino acids, I want to just emphasize the 2 kind of chemical literacy concepts that are needed to interpret these drawings. The first thing is we have to remember that we're always dealing with the Zwitter ion, right? I said in the beginning of this lesson that amino acids are typically found in a neutral environment and kind of standard water inside our cells. And it just turns out that the form of the amino acids we have are the forms where we have a positive charge nitrogen and a negative charge oxygen. And again, we're going to talk more about why is it that the oxygen becomes deprotonated and the nitrogen doesn't. That will be in another lesson. That's more of an advanced idea. But for now, just kind of trust me that amino acids are found in this state, in this Zwitter ion state, this mixed positive and negative ion form. All right, so we have our glycine, our alanine. Let's look at a few other amino acids that are in the same family, in this hydrocarbon side chain family. So what is this idea of side chain? You may already see what it means. The side chain is the chain of atoms, in this case mostly carbons, that kind of stick off the side of the amino acid. So you can kind of look at glycine. Glycine is the most simple amino acid. Alanine is the second most simple. It has a side chain of 1 carbon. Valine has this cool kind of V-shaped arrangement to its carbon side chain, right? It looks like a V there, just happens to be called valine. It's not named after the V shape, but just a nice way of remembering it. So the side chain of valine is 3 carbons. The side chain of leucine is 4 carbons. It's kind of got this kind of Y shape to it. The side chain of isoleucine is a little different. Isoleucine, by the way, is an isomer of leucine. Leucine has 4 carbons in the side chain, so does isoleucine, but it's in a different arrangement, different isomer. By the way, isoleucine has a second chiral carbon, but I'm not going to get into that. If you build it, and I hope you do build all these, you'll see that the third carbon, the carbon that's directly attached, the first carbon in the side chain, is itself a chiral carbon. I'm not indicating the chirality here, though. The next amino acid is a really cool one. It's a very special one. It's called proline. And this one, if you build it, and you have to build all these to really understand what they are, proline has a ringed side chain, the side chain that folds back on itself and it connects back to the nitrogen. And you'll notice that the nitrogen, how many bonds are on that nitrogen? There's still 4 bonds. It's just that one of the hydrogens is missing, we only have 2 hydrogens attached. All the others have 3. That's a very special feature of proline. Wow, okay, so let's kind of step back and pause for a minute. We are in the process of introducing the 20 amino acids that make up the proteins and enzymes that constitute living things on Earth. We know how many amino acids now? 1, 2, 3, 4, 5, 6 amino acids. These 6 amino acids have side chains that are mostly made of carbon. So honestly, before you go on to learn the other amino acids, I would stop here and spend some time building these, drawing them, and understanding what we're talking about here, and kind of slowing down, building each one, looking at the zwitterion aspect, looking at the fact that we have an extra proton in the nitrogen and we have a proton removed from the oxygen. Make sure you understand the chiral concept. Make sure that your side chain is pointing towards you, right? Because that's what that wedge means. That solid wedge means that when you build these, they should be coming towards you. So let's kind of quickly look at alanine for a second. Here's our pi mole structure of alanine. Hopefully you're building these in using the models as well. But here we see we have the left-handed form of alanine. We know it's the left-handed form because we have for the 3 high-ranking groups, we have the nitrogen is number 1. That's the higher-ranking carbon. That's the lower-ranking carbon. So the molecule is going to actually spin to the left here. This is the kind of alanine that we have in our bodies. So hopefully you're building structures like this and understanding. Oh, and the wedge idea, right? See how this wedge is kind of pointing towards us, whereas that hydrogen is pointing away from us. Make sure that you're building these correctly. It's really, really important in this unit to understand this idea of the acid-base nature of amino acids, but also the chirality, the chiral nature, the handed nature of amino acids. Now, something that might sound a little scary to people, but don't worry because everyone eventually learns to do this. You are supposed to memorize these. If you really want to be a chemist or you really want to be a biochemist, they know the structures of these, they memorize them. All of my students have memorized the 20 amino acids. We're starting here with just a few, but before we go into the rest, just focus on these, okay? These are the simpler ones. Spend even a whole day or maybe even a week, if you're a homeschool student, spend a week pondering these, drawing these, building them, practicing the names. Spend some time researching each one. You can look into that, where they got the names is very interesting too. This is college biochemistry right here. This is not something that's in AP chemistry even or other levels of chemistry. This is real college science. So at this point, the course starts to kind of slow down a bit because it becomes a little bit more intense and you have to memorize a few things. You have to kind of learn the ABCs of biochemistry here. We literally have an A, right? We have our A, we have our LV. We're literally learning the ABCs of life. So this takes a little bit of time. It's not something that, you're going to watch this video and, half an hour later, you're going to know all the amino acids. You have to spend some time focusing on these and learning them, drawing them, and building them. All right, so that said, we're going to go on and introduce the rest of the amino acids. And the way to learn these, like I said, is to draw them a lot and to build them a lot. And only then are we really capable of advancing on to the later units. I think that anyone who tries can do this. It just takes a little bit of concentration. It's just like learning the notes of the scale of a new instrument you're learning to play, or learning some new letters or new symbols in a language that you're learning. This is the next level, right? So the first level was learning the elements and how the elements act, right? Now we're learning the 20 amino acids. The 20 amino acids come together to make proteins, which is these grandiose structures of biology. So it's really important to learn your amino acids. Let's learn 2 more amino acids, serine and threonine. Serine looks a lot like alanine, doesn't it? The only difference is that it has an OH group on it. So if you have an alanine built, just put an OH on it, now you have serine. Threonine is a lot like serine, but it has an extra carbon. It has an extra CH3 group on it. And remember, there's hydrogens on these too, right? Don't ever forget to add the hydrogens. The threonine is a lot like serine. These are very related amino acids. Let's look at some more. Asparagine and glutamine. And yes, asparagine, The name comes from asparagus because these aspartic acid, which is another one we'll do in a second, was isolated from asparagus. What is the deal with the side chains here? We have a carbon with a double bonded oxygen, and an NH2 group. That's the side chain of asparagine. And we're going to learn what these side chains do in later lessons. What's the significance of all this later? Glutamine, what's the connection between glutamine and asparagine? One additional carbon. They're very, very related. The glutamine is like the big brother of asparagine, right? Just a longer version of it. Let's now do two more amino acids that are very similar to asparagine, aspartate and glutamate. Aspartate is the base form of aspartic acid, if you want to look up aspartic acid, and then glutamate is the base form, the deprotonated form of glutamic acid. Remember that those negative charged oxygens can actually be protonated. So that's another acidic group there. We're getting pretty complicated now. We're seeing how many acids can we have in a molecule. This molecule has 3 potential acids to it, right? Aspartate and glutamate. One thing that I should mention is that the one-letter codes are getting a little bit complicated. For the first line, the one-letter codes were pretty simple. But for the second line here, we have N, Q, D, E. Where do these letters come from? It's just that they ran out of letters, right? So glutamate can't be G because glycine was already G. Asparagine can't be A because alanine was already A. All right, same thing for aspartate and glutamate. So they just had to pick other letters, and there's different reasons why they pick those letters. But the one letter codes for these will be important when we explore pi mole structures of proteins. So don't worry too much about the one letter codes right now. Learn about the actual names of each of these amino acids and their structures. All right, so we're over halfway done learning the 20, or at least introducing the 20 amino acids. I don't expect anyone to have learned these as I'm talking. This is a kind of a long-term process. It should take a week or two to really let these structures settle in so that you memorize them, so that when we start looking at protein structure, it's very natural and you're very comfortable with what these amino acids and what their side chains look like. Here's 2 cool amino acids that are pretty unique in that they have sulfurs as part of their side chain, right? So the top one here is called cysteine. Cysteine looks a lot like alanine, doesn't it? But the only difference is that it has an SH on it. So you can use your yellow piece, your yellow atom piece, to show the SH. By the way, cysteine also looks a lot like serine, doesn't it? But instead of an OH, it has an SH. Methionine is a very important amino acid, and methionine has a long side chain of a carbon, a sulfur, and a carbon. Every protein starts with a methionine. So it's almost like a capital letter in the language of proteins. We'll talk more about that later. But methionine is quite a famous amino acid, so practice building that one as well. Let's look at 2 more amino acids. These amino acids have positively charged side chains, just like aspartate and glutamate have the base form of oxygen, which makes it a negatively charged side chain. The side chains of arginine and lysine have nitrogens, which we know nitrogens can accommodate up to 4 bonds, which when they have 4 bonds, they end up with a positive charge. So the side chains of arginine and lysine have these kinds of properties to them. These are pretty complicated side chains, right? The arginine has a carbon, a carbon, a carbon, and then this strange sort of arrangement of nitrogens and oxygens. That's called a guanidinium group, by the way, if you want to Google that or look more into it. But just for now, just appreciate the complexity, appreciate the fact that it's positively charged. Go ahead and build it. It'll help you understand it. Lysine, a little bit less complicated, actually. It's got a long side chain with a positively charged nitrogen at the end. Notice that the NH3, the normal one, that's part of the regular part of the amino acid, and there's another NH3 at the side chain. So this molecule has 2 NH3s in it. Just like aspartate has 2 negatively charged oxygens, same kind of idea. It's kind of a little bit symmetrical in a way. And then let's look at the remaining 4 amino acids, which are ringed amino acids. We're going to learn what am I talking about aromatic here. Aromaticity is another really important concept in chemistry and biochemistry, but this will come later. We'll learn about aromaticity later. But for now, just think of them as ringed side chains. So we have histidine, phenylalanine, tyrosine, and tryptophan, and they all have rings. Histidine has a ring that contains nitrogens. Phenylalanine has a ring that only has carbons. Tyrosine has a ring with 6 carbons that also has an OH group. And then tryptophan has a double ring, and one of the rings has a nitrogen in it. So these require a lot of atoms to build these. You probably can't build them all at once unless you have a lot of kits like me. But if you have just the student kit, you can probably build a couple of these at a time. But I really, really would suggest building these, drawing them. One cool thing to notice about them is that they're all related to alanine. They all have complex side chains except for glycine. But if you kind of ignore the ends of the side chains, they're all really alanine. So if you can build an alanine, if you can draw an alanine, if you remember the connections of the atoms, if you remember the chirality of it, you're off to a really good start. It's just a matter of adding the extra atoms to figure out the other, 19 or 18 amino acids. So why are we going to all this trouble to learning the 20 amino acids? Why am I making you do this crazy task? The point of doing this is because we are made of polymers of amino acids. We saw in our little plastic exercise where we made the polyethylene that we can have small monomers joining together to make polymers. Amino acids do the same thing, but they fold together, they connect together, and then they fold together into amazing elaborate structures that are also molecular machines. And these machines are the pieces that go into making life possible. So here's a close-up of one of those proteins, and we're going to understand more of what we're looking at here, but there's quite a lot going on here. But you may actually see some of the amino acids, and especially the side chains of the amino acids that we've already introduced. The amino acid at the top, if you have your amino acid sheet, spend a minute looking at this and try to figure out what that side chain is. It's a very long side chain. It has 3 nitrogens in it. There's only one amino acid that has a side chain like that, and that's arginine. So we can see that's just one example of where you would find an amino acid. That's arginine. And we see there's all these other like dotted line things. These are hydrogen bonds, which is part of the next unit. But I'm just trying to show you the amazing, where we're going here, these amazing structures that amino acids can create. Here's another close-up of a protein that has a few amino acids that you may recognize. We have some ringed amino acids, right? There's actually quite a few prolines here, which if you look at your amino acid sheet, you'll find them. In the bottom, we have a sulfur containing amino acid side chain. That's a methionine. We also have, looks like a asparagine in the center connecting to this water molecule. There's also a tyrosine in the top center. So all these elaborate structures that proteins are conforming to, they're all just made of amino acid polymers. Only those 20 amino acids go into making these structures. Now, one thing that I'd also recommend that you do is to explore full proteins and kind of zoom into them and try to find amino acid side chains. This only really works after you've looked at the amino acids for a while and kind of started to learn them. But if you zoom into this, anywhere you look, you'll find those 20 amino acids. We can already see those sulfurs there, right? Those sulfurs are part of either cysteine or methionine. And this is a great way to practice and kind of get a little bit more of a fun way of practicing your amino acids. Rather than just sitting and memorizing them, you can also kind of get a different skill looking at your proteins and finding amino acids there. So far there are 2 assignments. One is to learn the 20 amino acids, and the second is to practice identifying them, maybe by looking through the hemoglobin protein databank file and or other files and looking for the side chains to practice them. All right, so the ultimate real-life example of amino acids and proteins that's a living thing. Living things are mostly made of proteins. So if you wanted to ask someone like, why am I studying biochemistry? What's the purpose? What's the real life application? Living things are made of biomolecules. They're made of organic molecules and amino acids and proteins. So if you want to understand what living things are, you have to learn about amino acids. But there's some even kind of more direct connections to real life and everyday experience that we're going to go over here just before we end the lesson. I'm just going to show you some amino acids that are pretty famous. One amino acid that most people actually know by name is MSG, monosodium glutamate. Now, this is very similar to the glutamic acid molecule that we saw before, although it's the acid form. The side chain, right, it has an OH there, not an O minus, so that's the acid form. And we also see that there's a sodium that's connected to it. Now, we'll learn about how that sodium gets there, the idea of ionic bonds, but in a later unit. But this molecule here is an amino acid. It's monosodium glutamate. It has one sodium in it. That's why it's monosodium glutamate. And this is a really famous molecule. It's MSG. It's really famous for being in a lot of foods, including soy sauce, very famous for containing a lot of MSG. And things like cheese also contain a lot of MSG. The reason cheese has MSG in it is because cheese is made of protein, and the monosodium glutamate, the glutamic acid, was part of the protein. So it kind of makes sense that there's MSG. And the cool thing about MSG is that it's actually a flavor. It's the umami flavor. So there are certain flavors that we sense. We sense sweet, sour, salty, bitter. We also sense umami. That's the fifth flavor that we sense. And the molecule that creates this flavor is monosodium glutamate. So if you have a food that tastes kind of meaty or savory, it's because there's probably monosodium glutamate in that food. Here's another kind of interesting, sort of famous amino acid. Not an amino acid exactly, but something that's from an amino acid. So lysine was, remember that amino acid on the bottom left of our 20 amino acids table. Lysine is used to make proteins, but when an animal dies, something interesting happens to lysine. The right-hand carbon of the lysine gets removed by enzymes that are digesting and rotting the proteins away. That carbon leaves in the form of what molecule? CO2, right? So that molecule goes away, that part of the molecule goes away as a gas, leaves behind sort of like the side chain plus the nitrogen. That molecule that's left over is called cadaverin. Cadaverin, and that is the smell of rotting meat. All right? So when bacteria get at a protein in the meat, it removes the CO2 from the amino acid and leaves behind this double nitrogen containing molecule that is perceived by us as a really, really bad smell, which is why it's called cadaverin. And it's just kind of interesting that we are able to detect that rotting meat process by sensing the presence of cadaverin. So this may be the reason why meat smells bad to us, because it's kind of telling us that something's already been digesting this meat. Something's already rotting this meat. So it's not going to be something that we're going to want to get involved with eating. So this is just an interesting connection to amino acids. And if you ever passed a dead animal on the side of the road, you've smelled cadaverin, which is a product of lysine degradation. That's a cool thing, I think. Let's look at another amino acid degradation product of an amino acid that can turn into something else. This is histidine, which is one of the amino acids you're supposed to learn. It's one of the tricky ones. It has a kind of a tricky side chain to it. Histidine can also be changed. We can also take the carbon dioxide part off the histidine, and this is done by an enzyme, by a natural enzyme. It's not something that happens when something dies. It's part of a natural process inside an organism. And this gives us a molecule called histamine. It's not histidine anymore, it's histamine. And you may have heard of histamine because histamine is actually a hormone that gives us kind of an itchy sensation. It's the itchy molecule. If you've ever had an itch, from my understanding, it's because histamine has been released by your cells, and it gives that instinct to itch. And the reason people may know histamines is because a lot of medicines are antihistamines. They're molecules that the medicine, for example, Benadryl, stops this process, stops this enzyme conversion of histidine to histamine, and it's an anti-itch medicine, right? Because if you prevent histamine from forming, that means that you don't get an itch. And so if you have like poison ivy or an allergic reaction, you may take an antihistamine so that your body does not produce histamine anymore. Here are a few other amino acids that do other things, which are kind of cool and interesting: tyrosine and tryptophan. So these molecules make proteins, which is my main interest in this, and what we'll be studying in the other parts of the course. But tyrosine and tryptophan are really, really cool molecules because they also form hormones. They form very famous hormones like dopamine, adrenaline. You've probably heard of these. These give you different kinds of physiological or even mental responses, right? When your body turns tyrosine into adrenaline by kind of adding a few molecules here and there, right? There's not that a huge difference between tyrosine and these other molecules. They kind of just have some extra atoms added here and there. This gives molecules of completely different properties, and it gives molecules that do different things to our bodies. Tryptophan is interesting in that it produces serotonin, and further processing in the body can produce melatonin. And these molecules are important in sleep, right? And our mood and how we feel. So amino acids are not only connected to protein structures, but they're also literally connected to how we feel, which I think is really amazing to realize that molecules are involved in even this aspect of life. Now, if you're really into amino acids, you can memorize and draw and build models of these molecules as well. These are famous molecules. I've even seen people with tattoos or necklaces of serotonin or dopamine or other things like that. So these are molecules that are common even in popular culture. But for me, you don't have to memorize the structures of these. You do have to memorize the structures of the 20 amino acids, though, because going forward in the course, it's so important that you're at least familiar with them, especially some of the simpler ones. So that's my very strong suggestion before proceeding to lesson, it's 8 now, right? Learn your 20 amino acids, and we're going to have a great time going forward in the course. So thank you for joining me with this very long lesson, and take your time before going to the next one. Learn those 20 amino acids, and I'll see you in the next lesson.